Hello students, in this video lecture we are going to discuss about the DNA and the structures of DNA, right? What are the components of DNA, how DNA get composed of the different kinds of elements, and what are their types and what are the functions of this DNA. So that we are going to learn in this lecture video, right? So DNA, it's deoxyribose nucleic acids. It's a genetic material which are present in most of the living organisms. So regarding that we are going to learn. So DNA, it's a polymer of deoxyribose nucleic acid which found in the chromosomes as well as the mitochondrial DNA or mitochondrial DNA as well as the chloroplast DNA also, right? So they carries the genetic information, right? Whatever it will be get transferred from one generation to the next generation, right? So these DNA, the principal genetic material which are found in the most of the living organisms, right? So sometimes it's also called as a nucleic acid, and thus these are different complex molecules. They get together to form the structure of this DNA, right? So in the 1869, the scientist Frederick Mischer, in when he was just 22 year old, okay, he was a physician and he isolated from the pus cells. So he isolated the DNA from the pus cells and he just obtained from the discarded bandage, right, in the Franco-Prussian wars, and thus the accidentally he found the structure of this DNA, right? So there is a much more history regarding the discovery of the DNA. So we are not going to discuss regarding that. We will just discuss about the structure of the DNA, right? Deoxyribose nucleic acid, that is also called DNA. So the highly purified DNA, it's extracted from the wide different kinds of varieties from the variety of plants as well as animals, bacteria, viruses, right? So they has been found to be complex macromolecules. Macro means larger molecules, right? So and these are the polymeric chemical compounds which obtain contains the four kinds of smaller building blocks which are also called as a monomers, right? So that we are going to learn. So the components of the nucleic acid, that is DNA, is consist of three different components: nitrogen bases, sugar, and the phosphate groups, right? So these nitrogen bases which are found in the DNA is adenine, guanine, cytosine, and thymine, right? So these are the nitrogen bases, and sugar is a deoxyribose sugar which are found in the DNA, and the phosphate group obviously it's also present in the DNA, so that's why DNA is negatively charged because of this phosphate group, presence of this phosphate group, as well as DNA, it's chemically it's acidic in nature because of this phosphate groups, right? So we will going to learn about this in detail. So here you can see in these structures, right, you can find here the nitrogen base, sugar, right, as well as the phosphate group. So you can see here the nitrogen base. So there are different kinds of nitrogen bases, but generally we will just consider here nitrogen base. And see here, this is the sugar, right? Either if it is found in the DNA, it will be deoxyribose sugar, or it is found in the RNA, then it will be ribose sugar. Only the single difference is there in the deoxyribose and ribose sugar is that what I think you can see here the X. So this X letter denotes the presence of hydrogen in this sugar, then it will be a DNA. And if this X represent OH group, then it's RNA. Means there is a single difference between the DNA and the RNA structure, that is the presence or absence of oxygen at the second carbon number, right? So in this second carbon number you can see this X denote H in the DNA and X denote OH group in the RNA. So this is the nucleoside, right? So nucleoside is consist of the bases and sugar, and if I will add the phosphate group, attach the phosphate group to the sugar, then it consists of nucleotide, right? So this consists of the nucleotide. Nucleoside consists of bases, nitrogen bases and sugar only, while the nucleotide consists of nitrogen bases, sugar and the phosphate group, right? So here see one by one the basic structure of pyrimidine and the purines. So here you can see these are the pyrimidine consist of 6 carbon compound, okay, while purines they consist of the 9 carbon compound structure, right? So this is the basic structure of pyrimidine and purine. Then we will see in the next PPT the pyrimides. So this common structure of the pyrimidine is get varies into 3 different structure, that is cytosine, thymine and the uracil, okay. So you can observe the differences in their structures, right? So cytosine and the thymine, these are found in the DNA, while cytosine and uracil are found in the RNA. Means thymine is absent or restricted only in the DNA, while uracil is restricted to RNA structures only. So these are the pyrimidines, right? Then we'll see the purines. So this is the structure of purines. These are the double ring structure. Here you can see. So as compared to the pyrimidines, the purines are larger in size, as well as they are larger in the on the basis of molecular weight also. So here you can see this common structure get varies into 2 different kinds, that is adenine and guanine. So this adenine as well as guanine, they are found both in the DNA structure as well as RNA structures, okay. Then we will see the nomenclature of these nucleic acid components. The bases are of two types, purines and pyrimidines. The purines like the adenine, right? So adenine when it's present in the nucleoside form, it's adenosine, and when it phosphate group will attach, it will becomes the adenylate if it's found in the RNA. The deoxy adenosine, when it doesn't have the phosphate group, it will be called as a deoxy adenosine, and when the phosphate group will attach, it will becomes deoxyadenylate, and it's found in the DNA. Then guanine. So this guanine, it's commonly called as a guanosine when lacks the phosphate group, and when it will get attached to it, then it will becomes the guanylate, and guanylate found in the nucleic acids. Deoxyguanosine when it doesn't have phosphate group, and when phosphate group will attach, deoxyguanosine turn out into the deoxyguanylate, and it's found in the DNA only. So this is about the purine, right? Then we will talk about the pyrimidines. So pyrimidines, there are 3 different kinds of pyrimidines: cytosine, thymine and uracil. So cytosine, when it's in the presence in the form of nucleoside, it becomes the cytidine, and when phosphate group will attach, it will becomes the cytidylate, right? And it's found in the RNA only. When the deoxycytidine, when phosphate group will attach, it will become the deoxycytidylate, and thus it's called as a DNA. If I'm talking about the thymine, the nomenclature of this thymine, when it will be in the nucleoside form, it will be called as a thymidine, okay. And when the phosphate group will attach, it will becomes thymidylate, okay. and these are found only in the DNA only. Okay, and in case of the uracil, when it present in the nucleoside form, you will call it as a uridine, and phosphate group will attach and it will turn out into the nucleotides. It will become the uridylate, and it's found in the RNAs only, right? Then there is a very important formula which defines the amount of adenine and the guanine in the DNA structure. So that formula is given by the scientist Chargaff. That's why it is called as a Chargaff's rule, right? So this rule is also called as the equivalence rule, which is discovered by the scientist Chargaff in 1950. So it discovered the equivalence rule, which suggests that despite wide compositional variations exhibited by the different types of DNA, the total amount of purines equivalent to the total amount of pyrimidines, right? The amount of this adenine equaled the amount of thymine, right? And the amount of guanine equal to the amount of cytosine. So the Chargaff equivalence, the rule has been found to apply the almost universally different organisms, like in the viruses, bacteria, plants, and the animals, right? So however, the DNA isolated from the higher plants and the animals was in generally rich in the adenine and the thymine, and relatively poor in the guanine and cytosine, right? Whereas the DNA, which is isolated from the microorganisms like the viruses, bacteria, and the lower plants and animals, right, was in general rich in the generally rich in the guanine and the cytosine, and relatively poor in the adenine and thymine. Right? So these differences in the adenine thymine as to guanine cytosine ratio of the microorganisms and the higher organisms undoubtedly reflects the difference in the genetic information which is carried by the hereditary molecules, and also the phylogenetic, evolutionary, and the taxonomically significance of them, right? So that's why the scientist he postulated the formula that is called as a Chargaff's rules, right? So the he what he says, the amount of adenine is equals to the amount of thymine, and the amount of guanine is equals to the amount of cytosine, right? So he just failed to make the connection to the structures of this DNA and indicated that the DNA is symmetrical, right? So this is about the Chargaff's rule. Okay, then we will discuss about the DNA structure, right? So in the DNA molecule, the adjacent deoxyribose nucleic acids, they are joined in a chain by the phosphodiester bridge or the bonds with link the 5' carbon of the deoxyribose of one molecule to the another unit at the 3' carbon of the deoxyribose to the next mononucleotide units. So according to the Watson and Crick, right? So these are the two different scientists. The DNA molecules, they proves that the DNA molecule it consists of two such polynucleotide chains which are wrapped helically around each other with the sugar phosphate chain on the outside, right? And the purines and pyrimidines on the inside of the helix. The two polynucleotide strands, they are held together by the hydrogen bond, you know very well, right, in between the specific pairs of pyrimidines and pyrimidine, right? So we will see. So here you can see the structure of the primary structure of this DNA having these sequences right here. So here you can see the 5' end and the 3' end. So this 5' to 3' end, it shows the linkage in between this nucleotide, right? So this monomer, they get attached to form the structure of this RNA. So here you can see the structure, it is a single stranded structure. Okay, but just for your understanding, I am shown this PPT how there is a linkage in between these two different kinds of monomers of DNA, right? So there is a phosphodiester linkage in between the two different monomers, right, and which is run from the 5' end to the 3' end. So traditionally the DNA sequences is drawn from the 5' to the 3' end, and here also you can see the adenine, cytosine, guanine, thymine, adenine. So these sequences you have to read from 5' end to the 3' end. Okay, so a shorthand notation for this sequence is ACGTA. Okay, then we'll see this picture, right? You can see here, just imagine that this is the DNA. Okay, and this DNA is present in the its secondary structure, that is called the double helix structure. Okay, so this double helix structure, here you can see it just get coiled and coiled, right, to form the helical structure, as it is look like a helical in shape. That's why it's called double helix. And you can see, just imagine that these are the two different strands, so they are connected to each other by hydrogen bonds, right? So there is a linkage in between the two different strands, and that linkage is formed by the hydrogen bonds. So this is the secondary structure of the DNA, shows the two antiparallel polynucleotide chains just wrap or just wound around the same axis, right? So sugar and the phosphate chains, they wrap around the periphery. Okay, and the bases, that is the adenine, thymine, guanine and cytosine, occupies the core means inside area of the DNA structure, which forming the complementary adenine, thymine and guanine cytosine Watson-Crick base pairs. So Watson and Crick says that adenine always pair with the thymine and guanine will always pair with the cytosines, right? So the DNA double helix is held together mainly by the hydrogen bond, as I told you earlier. So this hydrogen bond, it's a chemical bond in which the hydrogen atom of one molecule is attracted to the electronegative atoms, especially these will be nitrogen, oxygen or fluorine atoms, usually of another molecules. So here you can see the linkage in between the hydrogen and oxygen, and this bond will be called as a hydrogen bond. Here you can see the two hydrogen bonds between the adenine thymine pairs and three hydrogen bonds between the guanine and cytosine pairs. Thus, they form the linkage in between the two different strands and forms the double stranded structure of DNA, right? So you can see here the diameter or the size in between the these structures, right? In the adenine and thymine, right? It shows the broader length of 11.1 angstrom unit. And if there is a linkage in between the guanine and cytosine, right, which are connected by the triple hydrogen bond, there is a 3 hydrogen bond, and this size shows the 10.8 angstrom unit. If you observe the hydrogen bonds of adenine and thymine, so first hydrogen bond shows the distance of 2.8 angstrom unit and another shows the 3 angstrom unit, while the bond in between the guanine and cytosine, the first hydrogen bond shows the width of 2.9 angstrom unit, second nitrogen bond shows the width of 3 angstrom unit, and another another hydrogen bond shows the width of 2.9 angstrom unit, right? The base stacking. So the bases in the DNA, they are planar and have a tendency to stack. Tendency to stack means tendency to get attached. So the major stacking forces like the hydrophobic interactions and the van der Waal forces or van der Waal interactions, they are responsible for the formation of this major stacking force. Okay. So there are different kinds of structural forms of DNA. Okay, found in the living organisms, right? These are the alternative forms of DNA double helices. So about 20 years after the discovery of the DNA, the double helix in the 1953, the DNA was thought have the same monotonous structure. Means in all the living organism of all the cells they have same kind of DNA with exactly 36 degree of the helical twist between it's just the adjacent base pairs. Okay, and the uniform helix geometry also. The subsequent experiment have shown the DNA is much more polymorphic. Poly means many. Okay, than the expected. Before that, what scientists have considered, there is a monotonous structure. But after the experimental evidences, they have said that DNA is a polymorphic in nature. So thus the DNA can be in the following types, what you can see in this PPT, right? In this slide. These different types of DNA describe the DNA molecules contains the right-handed helical coiling and has been called as a B form. C in the middle. Okay, B form or B DNA. It's a biologically important form of DNA, so that is commonly found in the nature. Okay, so in most living organisms, the B DNA is get formed. This shows the presence of B DNA. So this double helical structure of the DNA is found to exist in other alternative forms, such as at your left side ADNA, okay, and another side ZDNA. Okay, so this ADNA it's helix handedness, it shows the right handed structure, and Z DNA shows the left handed structure. The base pairs per turn means where the helical structure of the DNA get formed. So there the A form of DNA shows the 11 pairs, B form of DNA shows 10.4 pairs, and Z form shows 12. It means Z form of DNA is narrow structure as compared to other two. B is the broader structure. The rises per base pairs along the axis, which will be in the A DNA shows the 0.23 nanometer, while in the B DNA 0.34 nanometer, and while in the Z DNA 0.38 nanometer. The pitches found in the A DNA it's near about 2.46, in the B 3.40 nanometer, and in the Z 4.56 nanometer. The diameter of the this DNA, that is A DNA, is near about 2.55 nanometer, B form is 2.37, and Z form is 1.84. Means Z form of DNA is a narrow and elongated DNA as compared to B and A. So A is broader as compared to all these two. The conformation of the glycosidic bonds is antiparallel in the A form of DNA as like the B form of DNA also, but in the Z form the alternating anti and this synth form they shows the glycosidic bond in the Z form. Okay, the major groups are present in the A as well as B form of DNA, but absence in the Z form of DNA. Right? Then we'll see the normally hydrated DNA, B form of DNA. Here you can observe here the helical sense, they are right handed, and the base pairs almost perpendicular to the helic axis, which is 3.4 angstrom unit apart from each other, right? The one turn of the helix is, here you can see, they shows the 36 angstrom unit length, right, which is near about 10.4 base pairs. The major groove size is near about 12 angstrom unit, and minor grooves size is near about 12 angstrom unit, and the major grooves shows the 22 angstrom unit, right? So in the eukaryotic cells, so DNA is folded into the chromatin form, means DNA get condensed to form the structure of chromosomes. If you elongate single chromosome, right, if you stretch it, its length is near about 6 feet, right, means more than your height also, more than human normal height also, right? So how this elongated long DNA, it can be present in very smaller cells of smaller nucleus, and there are many chromosomes. Okay, so how this genetic material have to be get packed inside this small nucleus of small cells, right? So that's why this DNA get folded into the chromatin to be get condensed and packed inside the nucleus of the eukaryotic cells. So DNA is tertiary structure. We have seen the secondary structure, then we are going to learn about the tertiary structure of DNA. So DNA double helical structures, they coiled around the histone proteins. So there are proteins which are found in the structure of the DNA. So these proteins are called histone proteins. So DNA bound to these histone proteins, right? The nucleosomes, they will be called, which size is near about 10 nanometer. The nucleosomes, they contains 146 nucleotides in the single nucleosomes. So what are these nucleosomes, that we are going to see. So here you can see in this photograph any of the repeating globular subunits of chromatin, so that consists of a complex of DNA and the histone. So here you can see the DNA get wind up around these histone proteins, right? So that is called as the histone core of nucleosomes, and where the linkage in between these nucleosomes, so that is called as a linker DNA of nucleosomes, because it forms the linkage in between two nucleosomes. That's a linker DNA. So these histone proteins as well as the nucleosomes, their core structure is described in this photograph, right? So this is the molecular structure of this nucleosome. As we know that these histone proteins are octamer. Octamer means they consist of 8 different kind of subunits. So these 8 different kinds of subunits are S2A, S2B, okay, then S3 and H4, okay, and each copy is present there, means S2A having another copy, S2B having another copy, S3 having another copy, as well as S4 is also having another copy. So means there are how many total subunits? 8 subunits are there. So that's why it is called as a octamer, and on DNA is just get wind up around this octamer. So here you can see the compaction of the DNA into the eukaryotic chromosomes. So how the DNA get compact to form the structure of these chromosomes. So DNA is get beds on a string form of chromatin, right, which will forms the 30 nanometer fiber structure, and they form the loops, right, means they get coiled, which is near about 75,000 base pairs. Single loop having near about 75,000 base pairs, and they form such kind of loops. Many loops will get formed to form the structure of rosette. like the rose flower. Okay, so here you can see the one rosette which consists of the 6 loops, and it will be called as a rosette structure, right, of the nucleosome. At the center you will find the nuclear scaffold. Okay, so each rosette, which consists of six loops, will form the structure of nuclear scaffold, and it just gets coiled. So 30 different rosettes will just coiled to form the one coiling, right? And this coiling will get continue. So 10 different coils, they get together to form the structure of two chromatids. So in this way the DNA get condensed to form the structure of chromosomes. So here you can see the chromosomes. So in this, just for your better understanding what is meant by supercoiling or that, so I have shown this slide, right? We have the telephone and telephone wires. So sometimes what happens, this, because of the continuous use of this telephone, the telephone wire get coiled and get super coiled. So here you can see the coiling, and when this coiling will again get coiled, then it forms the super coiling. Then we will see the DNA melting and annealing. DNA can be melt, right? Melt means DNA structure can be get separated in the nitrogen bases, okay, in the sugar and phosphate group. Means these different molecules, they get together to form the structure of DNA, but they can be get separated also, right? So that we are going to see. So reversible renaturation and annealing of the DNA. So this is very important in case of the RNA replication, DNA replication. We will see in the next lecture what is meant by DNA replication. So the double helical DNA, right, it can be get denatured to convert it into the single stranded DNA, or single stranded DNA. Obviously it will be get together to by the hydrogen bond, it will be get linked and convert it again into double helical structure of DNA. So that is called as a annealing, right? So this separation of the strands as well as the association of the strands by the base pairing is also get formed, right? So that is called as the denaturation and annealing of DNA. Then we will see the melting point of DNA. So this graph shows the denaturation percentage of DNA and the temperature. So here you can see the temperature at the midpoint of the transition, it will be get, so it is noted by the Tm, right? That is the melting point of DNA. So as the temperature get increase, so DNA get melt. If it will be single stranded DNA, then what will happen? The melting point will be lower, and if DNA will be double stranded, then graph will be shifted towards the right side, right? Because temperature is getting increased, and this blue color graph shows the single stranded DNA, and red colored graph shows double stranded DNA. Okay, the Tm, the melting point of DNA, it depends on the content of guanosine and cytosine base pairs. So if the number of guanosine and cytosine base pairs will be get increased, then what will happen? The melting point of the DNA is also get increased. Okay, it also depends on the size of the DNA, pH of the environment, and the ionic strength. So here you can see the temperature, as in this graph you can see the temperature is getting increased. Okay, the guanosine cytosine concentration will be get increased, then the melting point also get increased. Okay, then finally we will see the functions of DNA and the summary of the DNA. So DNA, it consists of 4 nitrogen bases, adenine, guanine, cytosine and thymine, so which are get held in the linear arrays of phosphodiester bond through the 3' and 5' position of the adjacent deoxyribose moieties. So DNA is organized into two strands by the pairing of bases, that is adenine to thymine and guanine to cytosine on the complementary strands. So these strands they form a double helix around its central axis. The 3 into 10 raised to 9 basis pairs of DNA in the humans are organized into a haploid complement of 23 chromosomes. And you know that we are diploid living organism. Only our reproductive cells, that is sperms and egg cells, they have only the haploid cells, and rest of all cells are diploid cells. Means we have 46 chromosomes. The DNA it provides a template for its own replication. Means DNA can make the copy of itself like the Xerox machine. Okay, and thus the maintenance of the genotype and for the transcription of the roughly 30,000 human genes into a variety of RNA molecules. Okay, so today we will stop here. Thank you.